Hi, this is Mr. Arthur, and this is our part 2 of gene expression. So in the previous lesson, we talked about how DNA is transcribed into RNA, and we talked about the 3 different types of RNA. This lesson is going to focus about how that RNA is then translated into, first of all, a chain of amino acids, and then how that becomes a functioning protein. So to recap, remember a section of DNA which codes for a protein is called a gene. So that's why we call it gene expression. That gene is found in the nucleus. So that first stage of the DNA molecule unwinding or unzipping to expose the DNA bases is called transcription. And that transcription all happens in the nucleus, and it results in that mRNA being made. The first longer copy of mRNA is called the primary transcript. Before it leaves the nucleus, it shortens into the mature transcript as the introns are removed and the exons are joined together in RNA splicing. Today we're going to look at what happens here at the ribosome then. And this process is called translation. We translate the base sequence into the amino acid sequence. And that amino acid sequence forms a chain, which is then going to fold up into a working protein. Okay, so let's recap tRNA. This picture shows a tRNA. Remember, it's a single strand of RNA. It folds into a specific 3D shape due to complementary base pairs. It has a 3' end where a specific amino acid combined, and at the bottom there's 3 exposed bases which we call an anticodon. So remember, anticodon, think the third letter is a T, is part of the tRNA. Don't mix up the codon on the mRNA. So the tRNA is involved in the translation of mRNA into a polypeptide chain. That is the chain of amino acids, and that occurs at the ribosome. Remember that tRNA is called transfer RNA because it carries a specific amino acid. The tRNA is folded due to complementary base pairs, and that triplet of anticodons is key in explaining how translation works. So this picture shows translation. I know it's hard to see, but in the very top, you can see the first amino acid is green. That's been carried to the ribosome. Now we know it's the first one because the first codon in the mRNA is AUG, so it must bind the anticodon UAC. And that's the anticodon of that tRNA carrying MET, which is why that's the first one. Now, the ribosome slides along. The second codon is CCU, so it binds with the complementary codon GGA, which means that second yellow amino acid arrives, and the ribosome forms a peptide bond between these 2 amino acids to hold them in place. Again, the ribosome slides along. The first tRNA is now released because that amino acid in green is attached with the peptide bond to the yellow amino acid. So the tRNA can leave. The next exposed codon is ACU. So that binds with complementary base pairs to the anticodon UGA, which ensures the correct purple amino acid is now brought there by the tRNA. And again, the ribosome adds it to the polypeptide chain by using this peptide bond. This continues and continues and continues, so we get this growing polypeptide chain of amino acids held together by peptide bonds. So translation always begins with the first codon called a stop codon, and it will keep happening until you get, a start codon, and it will keep happening until you get to a stop codon, which stops translation. Remember, when we talk about anticodons, that's the tRNA, and the tRNA anticodons are complementary to the mRNA codons. That's the explanation as to how translation works. As well as saying they're complementary, try and say they join with complementary base pairing. The sequence of amino acids, they're joined together by bonds called peptide bonds. Remember, the tRNA is released after it's delivered its amino acid, and the amino acid is joined to the peptide chain, the polypeptide chain. Okay, this is quite common. The SUA give you a table of different amino acids, and it'll either show you the DNA triplets, the codons, or the anticodons that code for each one of them. Now, for the purposes of learning this, let's say this table shows us codons. So, which codon in the table would code for the amino acid triosine? So find TYR, and that will give you the 3 bases within the codon. Press play when you're ready for the answer. Okay, so up there, either answer is fine, UAU or UAC. Both of those codons will result in that blue amino acid, TYR. Let's try another one. Which amino acid would arrive if the mRNA codon was AAA? Press play again to get the answer. Okay, so AAA is down there, so that's going to give us the amino acid LYS. If the question asks for an amino acid, make sure you're writing these three unusual letters down, not one of the base combinations. That can be tricky because the bases are always in threes, and they tend to shorten the name of the amino acids to threes. Now, you don't need to know the names of the amino acids, they will always give you these, so you can just find it. So just identifying the information. The way I got to that one there then, ignore where the red is the first nucleotide labels on the left. So the first one's A, so I'll go down to the third row. Up the top it says second nucleotide, and again it's A, so I go to the third column, and then I come down where these two meet, and there's four options. Now the third nucleotide on the right is the third one down is A, so there we go, AAA, and that's how we find the one in the middle. So let's try another one for you to try this yourself. Which amino acid was the codon UUA code for? So the first U is up the top left, so we're going to be in the first row. Second letters are U, so we're going to be in the first column. So there's four options there and two amino acid options. The last one's an A, so it's the third one down. So our answer is LEU. Okay, what would the anticodon be on the tRNA which carries the protein proline to the ribosome? So the first thing to do is find proline. Then you have to remember that the table's codons, but the question is asking about anticodons, and that's really common for the SQA to show you one thing and ask about the other. So as long as you don't make the mistake of thinking that's the answer straight away, what we have to do is translate one of those codons into an anticodon. So if the first one's CCU, you would write GGA, CCC becomes GGG, CCA becomes GGU, because remember, anticodons are RNA, so no T's, and the last one is CCG, so that would be GGC. Any one of those answers would be correct in the red, as long as you remember to translate from the codon into the anticodon. Which amino acid would the codon UUC code for? So now it's giving you an anticodon, but the table's codons. So the first thing you have to do is change your anticodon into a codon. So to do that, UUC would become AAG. To find AAG, we would go here. A on the left, A at the top, and G on the right meets there. And the answer would be LYS. Okay, state a codon which would end the process of translation. So the KU knowledge is, well, it always ends with a stop codon. Up on the right, there's three red stop codons to choose from. So you could write down any of those, UAA, UAG, or UGA. Finally, it says, what would the final triplet in the gene coding for the protein be? Now it's asking about a gene, so we're looking at DNA, not RNA now. But we still know that the proteins end with a stop codon. So therefore, the end of the gene must be the DNA triplet, which ends up being a stop codon on the mRNA. So take our stop codons and just translate them back into DNA. So UAA becomes ATT, because remember DNA has thymine. UAG becomes ATC. And finally, UGA becomes ACT. So again, remembering that DNA has T's and just working back and forth. That was working back from a codon. If you were given an anticodon, you could change the anticodon into a codon and then change the codon into DNA. Okay, so just reading the question carefully is key to doing these types of questions. So let's look at the ribosome in action again here. So the first codon, if you were asked to identify it there, would be AUG. It's bound to the anticodon UAC, which ensures the correct amino acid, which is MET, arrives. Okay, now the second mRNA codon is GGA. The anticodon CCU has bound to the codon using complementary base pairing. That ensures the correct amino acid, in this case GLY, is added to the polypeptide chain. So that's an explanation of what's happening here if you're trying to write this into an answer for a question. You'll notice the amino acids are joined together with that purple bond, and that is a peptide bond. So now that MET is bound to GLY with a peptide bond, the first tRNA is free to leave. The third amino acid arrives now because the anticodon ACA is used complementary base pairs to bind to the codon UGU. Now, the AAG is blank, so you might be able to look at a diagram and see what happens next. Then you say, okay, well, AAG is complementary to UUC, so the next amino acid to be joined will be LYS. If you're asked the next one after that, well, go up and work yourself along in threes. The last code in this picture is CGA. So turn that into an anticodon would be GCU. Then we go back to our table and find out what amino acid would come next. So lots of problem solving involving these styles of questions, combining your knowledge of this and applying it to solve issues like what would the codon be, what would the anticodon be, what would the amino acid be. Now this is going to keep happening in the same way, each amino acid being bound to the next adjacent amino acid in the chain using peptide bonds, and that keeps happening until we reach the stop codon. When the stop codon is reached, translation is terminated, it ends. When it ends, that means that this polypeptide chain that we've made, down in the bottom left here, it's your chain of amino acids, is released from the ribosome. This video has also been put in the classroom to help show you this idea of protein synthesis. So, up the top right, so far what we've talked about is translation of a base sequence into an amino acid sequence. These amino acids are held together with peptide bonds, so we call that chain a polypeptide, but it's not yet a protein. To get to the protein, then our polypeptide chain has to fold into the correct 3D shape. So these are the words to use. The polypeptide folds, so it folds into a 3D shape of a protein. The reason that happens is, although the adjacent amino acids are held together with peptide bonds, certain amino acids are attracted to each other and they want to be closer together, so the chain ends up folding in on itself to allow those two amino acids which are attracted to bind using hydrogen bonds or other interactions. Other interactions, because we no longer teach you, you might see older questions that mention them, but for now I would just remember hydrogen bonds. That's the main one in the current course. Proteins have a large variety of shape. National 5 we said they were structural, hormonal, antibodies, receptors, so there's many enzymes. Each has a specific shape. That shape is down to the 3D folding. Remember that every gene is different, even though it has the same bases, because the order of the bases in the gene is different. Every protein is different because the order of the amino acids is different. That's really important because the order of the amino acids is what determines how it folds, and how it folds gives it shape, and the shape gives the function. So if there's a mistake in the amino acid sequence, it won't fold into the correct shape, and therefore the protein might not function. Now, it's not a case of one gene equals one mRNA equals one protein, because remember in the last lesson, alternative RNA splicing means that you can actually get different proteins from the same gene. To explain that, you get different mature transcripts from the same primary transcript depending on which exons are retained. If you get different exons, within that exon, you'll get different codons. Different codons mean they'll bind to different anticodons, so you get different tRNAs and therefore different amino acids being added to your polypeptide chain. If your polypeptide chains have different order of amino acids, they fold into different shapes, which means you've got two different proteins, both made probably out of the same amino acids, but a different order, so folding into a different shape to give them different functions. Finally, phenotype, National 5, we said it's your physical appearance. That is determined by the proteins produced. We did cellular differentiation. We said that stem cells become specialised through that process of differentiation because they express those genes which code for the proteins which are characteristic of that cell type. However, it's not just the genes, there's also environmental factors which influence your phenotype. Okay, you can pause on this. This is the SQA course notes. This is what the marking schemes were made of. So you can try and think about how to word this now you understand it using this language. So you can pause and have a look at that. This is the end of it. So again, you can pause. And then finally, I'll give you some basic questions to try. So try these 6 questions. And then when you're finished, you can pause the video, do it, go back to the video and see if you've written the right answer. Okay, good luck.